Hi, in this video we are going to talk about restriction enzymes. You can find written notes on what we're going to discuss today at medicinecanbeeasy.com. The link is also in the description. So what are you going to learn in this video? I'm going to explain what restriction enzymes are and how they work. We are also going to talk about why they are so useful and why we can use them in recombinant DNA technology. Okay, so let's start. Restriction enzymes, or restriction endonucleases, are enzymes produced by bacteria that recognize and cut specific nucleoside sequences in DNA, which we call recognition sequences or restriction sites. The enzymes cut both strands of DNA in the site by cleaving the phosphodiester bonds, the bonds between a phosphate group and a deoxyribose. They recognize short sequences of usually 4 to 6 base pairs. These recognition sequences are usually palindromic, which means that they read the same on the two complementary DNA strands in the 5' to 3' direction. When restriction enzymes cut DNA, some of them make staggered cuts. This way, we get what we call cohesive or sticky ends that are complementary to each other and can pair, anneal, with another complementary fragment cut by the same enzyme, forming hydrogen bonds between the complementary base pairs. DNA ligase can then join the fragments together and form recombinant DNA molecules. Recombinant DNA molecules are molecules produced by combining DNA from different sources. Restriction enzymes can also cut in the middle of its recognition sequence, producing blunt-ended fragments. The problem with restriction enzymes, which we've just described, is that their recognition sequences are very short, and they therefore produce thousands of DNA fragments. For that reason, geneticists have designed engineered nucleases that recognize longer DNA sequences. They consist of a restriction enzyme and a protein that is responsible for binding to a specific DNA sequence. An example of engineered nucleases are zinc-finger nucleases, which use a DNA binding domain called a zinc-finger and a restriction enzyme. Another example is a transcription activator-like effector nuclease, which contains a protein that binds to sequences in promoters. Now, why is all of this important? Restriction enzymes, as you can imagine, are very useful when we want to cut DNA into fragments. You also need to realize that each enzyme always cuts a DNA molecule at the same site. And for these reasons, restriction enzymes are used in recombinant DNA technology, molecular techniques used for locating, isolating, and changing DNA segments. It's called recombinant because it combines DNA that comes from different sources. Okay? So this is it. Thank you so much for watching. If you found this video helpful and you would like to watch more videos on genetics or medicine in general, please subscribe. Thank you again and see you soon.